Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. I'm very excited to bring you brand new episodes. To start, I had the pleasure of hosting Dr. Richard Premont. First, we discussed his career trajectory and then moved on to record a series of episodes in which he shared the history of the significant discoveries that shaped our field. I had such a great time chatting with Dr. Premont. I hope you'll have as much fun listening. Before we dive into this episode, I would like to take a moment to thank our Dr. GPCR Ecosystem partners, Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Please mark your calendar for our next symposium held on Friday, November 17th, on the topic of GPCRs in immuno-oncology. The symposium is free, but you must be a Dr. GPCR Ecosystem member, which is also free. For our last event of the year, we will be hosting a roundtable and discuss GPCRs and their role in immunology and oncology, and current therapeutic modalities in this context. The GPCR retreat is coming up November 2nd to the 4th. Although registration is closed, the organizers are looking for the event's next logo. Please visit gpcrretreat.org and get more details on the logo contest. Wouldn't it be fun to have your design associated with this iconic meeting? And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And today I'm very excited to have with me Dr. Richard Premont. Richard, welcome. Thank you for joining us today.
Well, thank you for having me. It's great to be here.
Very excited to, please.
No, just, you're always going about, the Quebecois. So my family background is Quebecois. So I pronounce it Premont, but up there they would say Premont.
See, I did not know that. So we can definitely dig into that. When you were a guest on the podcast when we did the series honoring Dr. Marc Caron, and I always have trouble when somebody has a French name. I had to think about how does, for example, Kathleen, his daughter, say his name and then use the same thing, because automatically it would come as Caron. That's fantastic. Thank you for being here. So let's start at the beginning. And would you please introduce yourself and tell us who you are and what do you do?
Sure. So my name is Richard Premont, and I'm a PhD who is actually still an associate professor. Have been basically forever, because I've never had a tenure track position, so that's kind of an oddity for your audience. I've always been in a medical environment where becoming a full professor, you have to pretty much be an MD. So I am very much a GPCR person, but I am much broader than that. I call myself a signal transduction researcher, because I take a very broad view of signal transduction by GPCRs and also how they talk with are integrated with other signaling systems. So I've been around a while. I got my first research job when I was still in high school, and have been working in science ever since.
Let's take a couple of steps back, because I think you said very interesting things. One, I don't think we've had someone with a long career who was still an associate professor, who's in a situation where you work with a lot of medical professionals. So I'm very interested in learning more about how did you get to where you are today. But also you mentioned, the distinction of signal transduction and the interest in signal transduction in the GPCR field. I really want to know, how did you get into that field? So let's start at the beginning. You mentioned having a French Quebecois descent.
Right.
Where were you born? Where did you study? And how did you get into science?
Sure. I'm just a plain old American. I mean, I was born in Springfield, Massachusetts, but my great-grandparents came from Quebec. Actually, it's kind of funny. The genealogy has been done, and the family comes from Ile d'Orléans, which is a little island in the St. Lawrence River right outside Quebec. They're very early. So the family's been in the New World for a very long time. But I'm just a plain old American, as that can. And I've always been interested in a lot of different things. I could easily have been a literature professor or many other things, but had very good teachers when I was in high school. particularly my biology teacher and my chemistry teacher. And they of course pushed me toward medicine, but I recognized very early that I'm interested in how things work. I'm interested in medicine per se, but I most certainly am not interested in practicing it. So over my career, I've always been in a medical environment because I like translational and that kinds of research, or research with a purpose, but not to the extent that I want to be the one with the stethoscope. Right.
Well, that's great. And a lot of guests that I had on had the medical profession in mind. And then very early on, they got into a lab environment and they realized, oh, actually, I'm way more comfortable working in the lab and asking questions and getting to understand how processes work. When did that happen for you? You mentioned having a good biology and chemistry teacher in high school. How did that influence your career?
I got very lucky. I've gotten very lucky in a lot of different ways along the way. But one of the things that very lucky is a friend of mine in high school. His father ran a laboratory in Houston at the MD Anderson Cancer Center. So when I was in high school, he was trying to get to med school, of course. So he would go volunteer in Dad's lab to get some experience working in a lab, and I got to do it as well, and had some very good experiences. I worked with some really people who turned out to be very fun, graduate students who turned out to be very phenomenal researchers later on, and got a very good start in general biological research. Also was working at a cancer hospital and really definitely knew I didn't want to be a physician. But that kind of grounding in hands-on research was something that was really cool. And it was something not a lot of people had access to at that time. Now there are a lot of established programs. At that time, that there really wasn't anything like that. So as an undergraduate, I went to Caltech, and there was no one my year except me who had any research experience walking in the door. So it was hard to do back in the day. So you had to know somebody basically. Yeah.
Yeah.
Um,
Yeah. Well, I'm glad
but
that
I loved it. It was a great experience, and have worked in research ever since.
I love it. I love the fact that, things have changed and very early on, there's these programs where you can have in high school, have this experience to spend some time in a lab, even if it's a short period of time, but you get exposed to this world and you
And
get to know
we're
it.
hosting undergraduates all the time, high school students frequently in the summer.
Yeah.
So yeah, it's a good thing to expose people to the career, because otherwise you don't know. It's one of those things, if you don't know somebody that does it, you don't know what people really do. So having a
Exactly.
hands-on experience really counts for a lot.
What did you work on in that lab?
Oh goodness. It's the only job I've ever had where I worked on something that was not involved with some kind of signaling. I've always been doing something endocrinological, but they worked on something called premature chromosome condensation, which was
Okay.
it's a weird thing where if you take a regular cell that is in any part of the cell cycle and fuse it together, like you're making a hybridoma, fuse it together with a cell that's in mitosis, you force the nucleus to undergo chromosome condensation, so the nucleus falls apart. And basically, they're working on cyclins before cyclins were invented, right? So you basically
Wow.
forcing the cells to change the state of the nucleus. They're trying to
Right.
understand. So it's interesting, kind of weird. But,
How cool is that? So, and then you went to Caltech. What was your degree? How did you, and you mentioned that you were the only person in your year who had lab experience. Did you look later on to get more lab experience? And when does the signal transduction come into your journey?
so yeah, so um, Caltech teaches you humility, because you think you're a big fish, and then you go there and there's always someone brighter than you. So but it's a very exciting environment. There's a lot of cool research going on. Um, I was also very lucky that whenever I would always go back to Houston. I came from Houston, so I was living in Houston and would go back every summer, one because my girlfriend was there, and not my wife, and would talk my way into jobs at the Houston Medical Center. So the next year I got a job with a guy named Grady Saunders, who was working on a growth hormone called human placental lactogen, um, which is a growth hormone family member that is made by the placenta. And so my job that summer was going to the waiting room and waiting for a baby to be born with my ice bucket and
Wow.
collecting the placenta. And the goal of what we're trying to do is basically we're going to do an RNase protection assay to measure the amounts of human placental lactogen RNA in term placentas. And it took me the whole summer because we collect the human tissue, dice it up, and then we would make the RNA using boiling phenol, using phenol that we redistilled in the lab because it didn't exist. You couldn't buy it, right?
Yeah.
And you had to make our own poly-RNA and little, like big drip columns. There were no kits. So it literally took the whole summer to do to
Yeah.
basically one experiment. So that was my introduction to molecular biology, but also got me very interested in endocrine function. And then every position I've had since then has been some hormone doing something somewhere.
Something.
Yes.
And they're very valuable and precise, and I think physiologically speaking, very interesting to think about and to work on. I love how you said that you talked yourself into a job. How did that happen?
And
I think
this is
that's important for people to understand. How do you do that?
right. I mean, these days, you just cold call. I mean, no one calls. I mean, kids these
Yeah.
days they don't make phone calls and text, but you cold contact somebody, you send them an email, right? There was no email, and snail mail. You didn't know an address of somebody to write to. There was
Yeah.
no internet to go look on a website to see what that address was. So if you didn't know somebody, you couldn't even write to them. So literally, I just went. I got there in the summer, and I just started walking down the hall, knocking on doors, until I found a job. So I
Wow.
needed a job, so I was gonna find one, and I did every summer during
But
college.
I think that's fantastic. And the reason why I wanted to stop and come back to it is because it shows determination.
Sure.
You
Yeah.
needed a job, you wanted a job, and you went and got it. Nowadays, it's an email. It may be a phone call in somebody's office if you
Mhm.
can find a phone number. But still, I think the vehicle is different, but the methodology, if you're looking for a job, if you're interested in
Sure.
joining a lab, nothing prevents you from reaching out. Worst case scenario, they're going to say, no,
Say
sorry, we
no.
don't have any space. Yeah.
Then you move on. Yeah.
Yeah. So you finished your degree at Caltech. What next?
So my girlfriend, then wife, was a year behind me in college, so I took a job as a research technician,
Mhm.
and I was working so I started working in a lab at Baylor College of Medicine, which is also in Houston. And they were working on G proteins. So that was how I got started in G proteins.
Mhm.
And it was very interesting experience because we were in the lab. We were trying to do things. The G proteins had just been shown to be heterotrimers. The lab had just purified GS, and then in purifying GS they found GI. So there's a lot of cool stuff going on. And we were also trying to identify purified glucagon receptors. So we were looking, basically interested in signaling in the liver and that's historical reasons
Yeah.
why that. Um, and then applied to graduate schools and got in. Both my wife and I got into graduate schools in Houston. So I was going to stay in Houston, and I decided to stay at Baylor. And I did look around doing other rotations, but I already had a year head start on a
Yeah.
project, so that was going to be hard to beat. So I stayed, and I've been now in GPCRs ever since.
Was that
Uh,
the first time when you got exposed to the GPCR world?
well, so I mean, I knew a lot about them from undergraduate
Mhm.
because one, we used the Stryer biochemistry textbook, and Stryer was a visual person, so it was all very heavily emphasizing visual transduction. And also, I took a couple of courses in visual transduction as an undergraduate. But also, we were lucky that there was a guy named Mel Simon who had just moved to Caltech like my senior year.
Mhm.
And he's later on, and he was teaching a class as well. And later on, he's the one that did all the cloning that found GQ and G11 and G12 and G13 and all of those. So he was very interested in G proteins as well. So I got a good, didn't do any research on them, but I had a good
Yeah.
handle on them going
Yeah.
into school. Yeah.
Yeah. So you kind of fell into it
Yeah,
to
yeah.
some extent, and then stayed in the in that field. So graduate school, working on glucagon, and GS has been cloned, GI has been cloned. I mean, it's so interesting because okay, there's when I went to undergrad, Michel Bouvier was teaching this class about signal transduction, and these things were, the way they were taught was like, we've always known this and this is done. And I think now is the good time to mention to the audience that you and I are going to be collaborating on a series of podcast episodes where we're going to be talking about the history of the field. So I can't wait to hear a little bit more about life before having discovered and
Mhm.
cloned these G proteins and GPCRs and the signaling effectors and what are the stories behind the scenes. So we're not going to talk about it today. I just want to mention it so that the audience can get ready for the next couple of episodes.
There's a lot of cool stuff there, and I was lucky to kind of land in the right labs at just kind of the right moments. I remember as a
Yeah.
graduate student, starting a manila folder with the beta receptor cloning paper. It was like receptor cloning is what I labeled it, and that one file got thicker and thicker and thicker over time. But yeah,
Yeah, yeah. Until you needed a second file, and at some
and
point,
suddenly you have your roar of, yeah. And now we have PDF, so we don't have.
exactly, you have different types of files. So graduate school, you're still and you're in that GPCR world and G protein world. What next after graduate school?
So my graduate experience was also very weird because I started at Baylor in the cell biology program, and then my boss decided to take a job in New York City. And I had the choice of either starting over again because I hadn't done my qualifying exam yet, which was then like 2.5 years into the program. I was only a year into the program, but I'd already been in the lab beforehand, so my project was pretty advanced. Or moving with him. So I ended up moving. So I ended up getting my degree from the City University of New York, working at Mount Sinai Hospital, New York City.
Wow.
I was in New York for several years. And while I was there, I had to basically restart all my classes again. I
Oh God.
had taken, biochemistry and histology and cell biology back at Baylor. And then when I moved to New York City, suddenly I had to take physiology and pharmacology. And this is all with the medical students.
Mhm.
So in the end, basically, I've taken all the medical classes,
Okay.
except, I never did human dissection. But other than that, pretty much I've taken all the medical classes,
Yeah.
plus a double lump of graduate classes. So, which is annoying because my wife is like, "You're going to finish it. You're going to finish it." But I have a very broad background, so that helps a lot in assessing other people's science or assessing, what do you want to work on? What's a cool thing? So, you win some, lose some. But I think
Yeah.
it was a good experience to be involved in that. And what I was working on was basically, so people nowadays they make induced pluripotent stem cells and they try and make hepatocytes, and
Yeah.
they say they can do it because they get albumin's expressed a little bit, whatever. But the problem with hepatocytes in particular, if you take hepatocytes out of the liver, they immediately stop being an hepatocyte, something called tissue specific extinction. They de-differentiate immediately. So one system where that doesn't happen is in chickens. So if you take baby chicks, and take hepatocytes out of their liver, they stay very much hepatocytes for over a week. So I was looking at the glucagon stimulated adenyl cyclase system in chick hepatocytes, and looking at how the receptor is regulated, so its desensitization, but also identified that there are feedback mechanisms that affect the G protein and also adenyl cyclase. So kind of my career has always been focused on the whole system, right from the beginning. So looking at how the dynamics of everything changes, and it's not just the receptor and GRKs and what have you, it's the whole system is changing. Yeah.
Yeah. And I think that's very unique, especially that early on. And the reason I'm saying this is because back in the day, we didn't have the tools we have today.
Right.
And so if you wanted to work on something specific, most people were focused on, a G protein or GPCR and not look at an entire system. And even now, I don't think we have all the tools that we need to be able to focus on a system or on an organ or on a total organism. I think we, at least from what I've been exposed to, there is this need to take a step back and try and look at, okay, we identified a signaling pathway, we know what's going on in a hex cell. How does that actually happen in a cell that endogenously expresses
Right.
those proteins? And what does that have to do with the entire function of an organ and an organism?
Absolutely. Yeah. I think that's something, I've always tried to be involved in science where people have a larger, more integrative look, which is
Yeah.
we had met actually doing the Mark Carone tribute, and one of the things that came out in that is, like, everyone was very impressed that Mark always had this very holistic view of how to do research, and
Yeah.
that's one of the reasons that I ended up at Duke later. Was, I appreciated that because my background was also looking that kind of a way. You can think of the receptors as a light switch, right? The ligand's there, the ligand's not there, it's on, it's off, but it's not really true. It's more like a dimmer switch where it's on,
Yeah.
you kind of tune it. So yeah, that's basically what I learned as a graduate student, is that the receptors are very sensitive to their initial conditions, and that the signaling depends on the state of the whole cell.
Yeah, of the entire environment. And I
And
think we think about it, maybe I'm generalizing, but a lot of people think about it as, receptor G protein, ligand receptor G protein, GRK, beta arrestin, and something else. But then we forget the lipids, we forget other GPCRs, we forget other proteins that are expressed in the membrane, and all of those things.
Absolutely. Absolutely. So I mean, if you, kind of as an analogy, right? If you and I are having a conversation, so you say something, I'm attending to you. But if we're at a party, there are lots of people talking. It's very hard for us to focus on one person because there's some other loud person off over there that distracts your attention. But cells listen to all those signals all the time, and they always do the right thing. So that's really fascinating. How do cells listen and integrate all that information to do the right thing? And they're not cells in a dish where you've serum starved it, and now you're adding a ton of your one ligand.
Yeah.
That little bit of ligand in a natural environment. There's a really famous video of a neutrophil following a bacteria through a plate of blood.
Yeah.
The amount of material given off by that bacteria is minuscule, but that cell sure as heck can note it and follow it.
Yeah.
So it's an amazing system how signaling can really work in the real world, not just in our artificial HEK cells.
Absolutely. I always find it fascinating. I feel like this, yeah, I just find it fascinating. You mentioned, being at a party and having all that signal, all that noise, and trying to focus on one conversation. I'm thinking here about, and a lot of these are learned skills, but I'm thinking about children who are tiny, and then there are things that automatically come to them because they can learn it and they can figure it out just by seeing it, and then the brain registers, and just to register that information and to learn and to respond to that information, I can only imagine the number of signaling events that need to happen. And when I think about hormones, for example, I mean, a little bit of a hormone the signaling induced by that goes such a long way, and it's instantaneous. How fast and how precise and how amazing that is. I just find it very fascinating.
Yep, that's why I like endocrinology.
So you were in New York at Mount Sinai. What next?
I was lucky and had an NSF predoctoral fellowship, which, when I started it, I was in Houston. It made me the most highly paid graduate student in the department, and then I moved to New York City, and it made me the lowest paid.
Yes, I was going to ask you.
Well, but one nice thing of having some of these fellowships is that I had my own pot of money for travel. So I was able to go, as a graduate student, I was able to pick the meeting I wanted to go to, and I just go to it. So one year I got to go to a meeting in the Netherlands. There was a very small meeting of like 30 people, sponsored by NATO. It was chemotaxis, basically. So there are people there like Peter Devriotes, but Mark Carone also happened to go to the meeting. So I met him there, And he's just spent the whole time trying to convince me, hey, you need to come to Duke when when you finish. So it's kind of really no question when I finished my graduate school. I went to Lefkowitz Caron land
Yeah.
at Duke. And once I was there, since I'd already been doing kind of receptor desensitization, they had just cloned GRK2, which at the time they called BARC, the beta adrenergic receptor. Yes. And they had just cloned the barrestins or the beta arrestins. And I was like, well, I want to do something, I appreciate what they're doing, with the molecular level of desensitization, but I want to do something different. So I probably, Bob always has a very tight focus on the beta 2 adrenergic receptor, as everyone seems, is pretty much aware. And I convinced him, let's try something really wacky. So I convinced him, let's try and clone tastant receptors. We know they're GPCRs. The olfactory receptors had just been cloned. It was a really big splash. So let's give it a try. So he said, yeah, okay, sure. So I actually started to do that. And while I was doing that, I was basically using degenerate PCR, which is kind of a new technique at the time, and identified several receptors, none of which turned out to be the taste receptors, because basically I was looking for class A or class B, so beta receptor like or glucagon secretin receptor like, and they're not. They're a different family that hadn't been identified at that time. But while I was doing that, I was also playing with other things. So I as a graduate student, I had cloned adenyl cyclases AC5 and AC6. So I thought, well, let me just see what taste buds have, and I just did degenerate PCR for adenyl cyclases and arrestins and GRKs and phospholipase C betas, and just what do they have? Is there anything specific? Because people are starting to find visual pathway and there's gusteucin, the G protein from the taste buds had been identified already. Are there other signaling components that are specific to taste buds? And never found any. But what I did find was a new GRK. And one of those things is a mistake or an accident that kind of changes your life. So here I am trying to clone taste receptors, and I had some receptor pieces and don't know what they are. They're just clearly a GPCR. And I found this bit of a new GRK. And back in those days, you did screening of clone libraries where you added a virus containing an individual clone to a plate of bacteria to get a little pole, a little colony, and then you'd lift those up, and then you'd expose that filter that you lifted it on to radioactive DNA probe. So I cut restriction digest. I cut out the receptor probe band to run it on a gel to purify it so I could label it. And on the same gel, I ran the GRK one,
Wow.
which was don't do it. I mean, no one does it anymore. You don't do this anymore, but don't do it. But I did that, and you think you're purifying two separate bands that are completely different. So I radio labeled the receptor probe, did a screen, found a bunch of colonies, really happy, and then I start sequencing them. None of them sequenced with, so I had primers based on the receptor sequence, and nothing was positive, nothing. So you sequence the ends of the clones and just do a search. It's like, what is this? It was a GRK. So that's any amount of carryover.
Yeah.
In the probe DNA, the GRK was very highly expressed. Again, it's not taste specific; it's everywhere. Turned out to be GRK5. So when after some time, we found a lot of receptors, and none of them by in situ hybridization, none of them were in the taste buds. I just said, enough of that. But by that time, I identified GRK5 and cloned it accidentally. So we'd sequenced it, and I just started, characterizing GRK5, and that's how I got involved with identifying the GRK4, 5, 6 family, which I stuck with for a long time. So, we cloned the cDNAs, we, cloned the genes, made the knockouts, all of that,
The whole
that whole family, which was a lot of fun. But not what I was planning to do at the beginning. So
yeah. Well, this is all about following where the science takes you. And although it was out of a little mistake, but at the same time, I think it was an important series of events and important discoveries.
Yeah. That's why I always recommend, if you're a postdoc somewhere, the boss is going to set you to one project, always have two. One is, pie in the sky. If it works, it's a home run kind of experiment. And the other one, it should be something that you're pretty certain will work, but it's still meaningful.
Yeah.
And if you're lucky, you get lucky. And if you don't get lucky, you have, you have fallback.
Yeah.
I think you talk about aha moments, and that was one, it was kind of a, an aha moment / wake up call. It's like, well, maybe I need to be thinking more about, molecular mechanisms of desensitization rather than the taste and receptors.
Yeah.
And um,
Where did the idea of working on the taste and receptors and taste buds come from? I mean, the motivation behind it.
so, I mean, I already knew, everyone knew that they were at least, sweet and bitter or G protein mediated. Sour was always thought to
Yeah.
be a
Yeah.
channel. Um, and when I was just about finished with graduate school, the olfactory receptor paper came out. And if you want to really amuse yourself, look, pull up a copy of that paper and look at the dates. It was accepted, it was submitted to Cell, and it was accepted to Cell, and it was printed within 2 weeks.
Wow.
So when they want to move fast, they can move very fast. So it made, so that just an indication, it made a splash. I mean, it was a big deal. Um, so thought, wow, somebody's going to do that for taste, and it might as well be me.
Yeah.
But it didn't work out that way. So
Well, there was something else in the plans for you
yeah, well,
on
that's
that end.
yeah. So
Wow. You mentioned having worked in Lefkowitz and Corona Lab. You mentioned doing the knockouts. Um, my question is around, so you went and characterized, cloned, got the cDNA, did the knockouts. What are some of the significant things that you found or discoveries that you made during your time in the Corona and Lefkowitz labs?
so I mean, so basically the mechanism for the GRKs is basically conserved. That is that there are basically three kinds of proteins that the receptor talks to: the G proteins, the GRKs, and the arrestins. And in each case, the receptor conformation affects the target conformation and vice versa, right?
Yeah.
So one of the things we showed is that for GRK4, 5, and 6, they also are activated by directly, with the activated conformation of the receptor activates the kinase activity. But unlike GRK2, at the time I arrived, that was right when they were showing that the beta gamma subunits activate GRK2, but GRK4, 5, and 6 do not bind beta gamma subunits. They're completely insensitive to it. But what's different about them is they have a various mechanisms by which they attach to the plasma membrane. So they're kind of constitutively at the plasma membrane. And in part for that reason, they have a high basal activity. So they're able to phosphorylate receptors even when they're not activated. In fact, they can phosphorylate receptors when they're bound to antagonists. Not so well, but they can.
Yeah.
So that kind of, their role is a little bit different. They're kind of more, they're able to stimulate, phosphorylation in response to agonists, but they also are involved in kind of a basal level somehow. Yeah.
Interesting. All right. I asked this from all of our guests, and it doesn't have to be a GPCR. What is your favorite GPCR or signal transduction molecule?
I have a few favorites for different reasons. So I mean, there's some, I don't know, some joy in, those of us of that generation who actually identified a thing that now you just go and the genome's there. You want to know something's there, it's there. You might find a new function, and that's great. And we do try to do that all the time. Find new functions for enzymes, but finding something completely new was a really cool thing. And going, so like Brian Kobilka, right? So he had cloned the beta receptor. He didn't purify it, right? But he cloned it, and then he went through and he made the knockouts. He got the crystal structure, right? So that's a kind of, there's a satisfaction in kind of carrying it through to that level. So in my case, I cloned adenyl cyclase 5, and we're able to show how it functioned. And it turns out, there is a foundation out there for kids that have knockouts.
Wow.
So it's like,
Wow.
I just kind of like, wow, something I found is,
Mm-hmm.
it really impacts people.
Yeah.
So when we were cloning GRK4, so GRK4 had been identified actually as a gene that was next to the Huntington's locus. It was Huntington's is interesting transcript 15, IT15, GRK4, IT11.
Wow.
But we had we recloned it, found splice variants, blah blah blah. It turns out that there are polymorphisms, in GRK4, lots of them, and I found them right from the beginning because we pulled sequences from several different libraries, and in our first paper, there's all these polymorphisms. P.S. Those polymorphisms have effects on people, right? They're disease. So that's really, it's cool, interesting, and
It
kind
is.
of satisfying to be involved in your little part of that.
Yeah.
So yeah, so dental cyclase 5, GRK4. I mean, the beta receptor, just as much as I try and get away from it, we keep on. It's the gift that keeps on giving. There's always some new wrinkle. So I mean, we just had a paper, a year ago on a completely novel way that it's regulated. Nobody had realized before. So yeah, as a receptor, I'd say the beta 2 receptor.
Yeah. Yeah, I mean,
Lots of favorites because
it's hard, but that's why I said it can be a GPCR, it can be any other signal transduction molecule, and it can be a family or a group of molecules. I think the most extreme or most interesting response I got was from Andrew Tobin, who had a phosphorylation site as a favorite site. I don't remember which one it was, but I thought it was really an interesting answer. Great. All right. So you're in the Caron/Lefkowitz lab. You have identified, cloned, and made knockouts of several GRKs. What next?
So basically when we started making knockout mice, suddenly I had to learn physiology. And that was something that the Caron lab was very good at. So I spent a lot of time, spending a lot of time with a lot of the postdocs in his lab, actually learning how to do physiological and even behavioral experiments, so that in the end, he's kind of famous for having had a behavioral facility in his lab.
Yeah.
Years later, I had one too, for the thing that I was doing, because I liked that model of going from understanding a molecule to understanding its function, and the only way to do that is to actually have that kind of stuff. So I spent a lot of time learning various kinds of physiology in order to assess how are the GRKs involved in regulating receptor functions. And it turns out that there's in a lot of cases there's a lot of redundancy. So one GRK knockout, you don't see a lot of phenotype, and occasionally you do, but not always. And it gets to be a bit of a slog trying to tease out. So you basically kind of hop from system to system to find the one that gives you the most robust phenotype. Yeah.
How interesting. You mentioned Mark's lab, and I think one take-home message that came out of the podcast series is that he was one of the first people at that time in an academic system that had what you can characterize as a pipeline, where he went, really from the molecule, from the protein to the animal to the behavioral.
Mm-hmm.
And there was this feedback loop, because obviously you have an idea about a project, but that's science. You go through the experiments and then the data takes you somewhere else. So it just, flourished from there.
Yep. So,
please go ahead.
so the other thing I did was, people had started looking at beta arrestins as signaling molecules in their own right. Mm-hmm. And I got interested in the idea that the GRKs also might be signaling molecules in their own right, either by phosphorylating other targets in response to receptor activation, or by binding to things. So I started looking for things that interacted with GRKs, found a couple of interesting proteins, and then kind of worked on them quite extensively after that. So that we call the GIT proteins, which is the GRK interactors, which turns out to be part of a complex of proteins that do everything. I mean, they're involved in so many things. It's crazy, but they really kind of scratch my itch for understanding how signals are integrated, because they interact with GRKs, they interact with the PAC kinases, so the P21 activated protein kinases. They are in a complex, so they are ARF gaps. So they regular GPase activating proteins for the small G protein ARF that interacts with a protein that's a guanine nucleotide exchange factor for the Rho family. So it's like a place where all these signaling complexes are all together at one time. It was really kind of fascinating to.
Yeah, this is where your image that you mentioned in the beginning comes into mind, where you're at a party, you're trying to have one conversation, and then everybody is talking around you. And what you just described with all of these signaling proteins is that's the image that came into my mind. Is every protein knows its role, but then has to be specific and precise and act at the right time in spite of everything else that's going on around it.
Well, I think you've had Stuart Maudsley on a few times before.
Yes, I have.
I stayed at Duke for a long time, so everybody that went through the Lefkowitz and Corona labs, I know them for 20 years. But yeah, I know Stuart very well. And he got me started thinking about this, and that is, if you think about proteins by analogy to like a social network, right? So there are clusters of people that all know each other, and then there are other clusters of people that don't know each other, and they're connected to each other by kind of loose connections, nodes. And so the node proteins are the ones that are kind of interesting because they allow these things that are happening within one complex to talk to things that are happening in another complex. And that's kind of what these proteins seem to do, is they're in this in-between state where they can listen to many conversations through different, different signaling systems and integrate them together.
Yeah, and Stuart has spent a lot of time working on Git too.
Yes.
If I remember correctly. Yeah, we've had great conversations, and he came to present multiple times and different Dr. GPCR events. So really phenomenal. It's really hard to stop talking to Stuart because he has all these very brilliant ideas, and you can have a really in-depth conversation with Stuart, as you as well know. All right, so you were in the Quran lab with the Lefkowitz lab, had the opportunity to work on different projects, learned a little bit more about physiology. Then what happened next in your career?
Yeah, so then I started looking around for jobs, and so I have a different attitude than a lot of scientists. So science is a career where people jump around from place to place to place, and it seems the more you jump, the better you do. But by then I had a family, and I really didn't. I know a lot of people that grew up that they either call them preachers' kids or military brats. People that move every 3-4 years, and I think it's not good for kids to grow up that way. So I said I'm not going to do that. I grew up in one place till I was 15, and then I wanted to have my kids to have that. So I didn't want to move a lot of times. And an opportunity came up to stay at Duke in a medical department in gastroenterology. So I took it, and I was there, for another 20 years almost.
Wow.
So I just, yeah. But I think, in looking, I mean, when I was looking for, even looking at original graduate schools, I didn't intend to go to New York City. Even then, we're poorly paid, right? For the level of education and the effort that we put in. So I'm not one who's going to go out of my way I'm not going to move to San Francisco. You can't pay me enough. I'd rather be relatively well off in a not so expensive place than feeling desperately poor in an expensive place. So since I was already settled in, we picked Durham because it was a place that was affordable to live at the time compared to other places. I was, thinking about, well, maybe I could go to, Seattle. There was a lab I was interested in. There were a couple of other places, and even then they were crazy expensive.
Yeah.
So that's a consideration I don't think you talk about too much, but it is a concern in
it is.
Career. Yeah.
It's an absolutely valid concern. I think once you have a family, you're not alone anymore. You have not only a spouse, but you have children. And you're right. I moved so many times, I stopped counting. And that's why my husband and I said, okay, Boston area, it's the Boston area. We're not moving. If we can help it, we're not moving.
Huh?
And because it's important to have stability for the children. It took us, I don't know about you with the moves, but it took us once you're established in a city, it takes 2, 3, 4 years until you actually build those relationships around you, that support system, with all the birthday parties and the different parents and all of that. And that's very important to have. And every time you move, you start from scratch. And you make another great point with the money considerations, because you're right. I think postdocs and PhD students are not paid enough for the time and effort and the energy they put in. And it's very hard to live in New York City or, Seattle, San Francisco, and even here in the Boston area. So it's something that needs to be taken into account. I'm not saying don't go to these places. I'm just saying think about what are your priorities as a person. Can you afford it? Yes or no? And what's the goal? What's the end goal?
Yeah. I mean, I enjoyed being in New York City. It was a great experience.
Yeah.
Didn't have kids at the time, so it was that it was okay from that point of view.
Yeah.
But yeah, it gets tough.
It
And
does.
I also had it modeled, right? So I was working in the Lefferts and Carone lab, and they, Bob is celebrating his 80th birthday this year. He's been there 50 years, 50 years.
I see.
And Mark had been there, 40 some. So they're kind of modeling, you can do good science staying in a place
Yes.
without having to jump around.
Yeah, no, absolutely. Absolutely. I saw the article that actually Stuart Motley had shared. It's published on Duke's website. It's going to be in our next week, Dr. GPCR Weekly News, about Bob's 50 years at Duke, which
It'll
is
be
big.
it'll be a fun time.
Yeah, no, agreed. And now you spent 20 years at Duke. Now you're in Cleveland. Tell us a little bit more about that transition.
Yes. So one thing about being in a place for a time is you kind of, those collaborations that you are going to have, you have, and, other things, they're just not going to happen. You can't force them. I had an opportunity to come here and my kids were grown enough. It's like, okay, so let's try something different. The reason is because although I'm sitting here at Case Western Reserve University, I don't actually work for Case. I work for something called the Harrington Discovery Institute. And what it is literally it's unique. It's an institution where they raise money by philanthropy and give away grants to people who are doing research that is aiming very much to be drugs. So you've discovered something, you think you've identified a drug target, you can apply to us for a grant. And we'll give you a grant, but we don't just give you a grant. We also have a team of people that gives you advice in how to position yourself for intellectual property, how to start a company, how to get an FDA, investigational new drug application approved, how to license to pharma. Really trying to, all the things that you would need in like a startup incubator that you might have to pay for if you started a company, we give them for free to help them get over, the valley of death where, drugs go to die. So we're trying to pick the best projects, have the best hope of actually turning into a marketable drug, and then giving them all the advice they need to, if they want to start a company, if that's their dream, we'll help them do that. If they want to license it to somebody else, we'll help them do that. If they want to go it alone for a while and try and do their own clinical trials, we'll help them do that. So really nobody else does this. So it's really kind of a remarkable place to be because, it gets to the other side of my skill set, which is basically having very broad interest and a broad background. So I see applications and we're helping people that are doing all, everything from, medical devices to all drugs for GPCRs, but drugs for all kinds of other targets. So it's a very neat environment. There's a lot of cool stuff going on, and it's it's that I'm not in industry, right? So I'm not actually going to help somebody get the drug approved, but I'm helping somebody that's on that path. So one degree removed from that satisfaction of getting a drug out.
Yeah, but still you have something to do with it in the sense that you get to see the science. You get to pick the best project and you get to give advice to give a higher chance for those potential drugs or drug targets to be used later on and end up helping people, which I think is really fantastic. I'd really love to learn more about this organization that you work at. Maybe we can have a separate chat about it because I think it's important. Now, I mean, a lot of people and I'm thinking here about myself when I was working in the lab. You're working in the lab, your focus is narrowed on your little project because you have to graduate or you have to publish the paper so that you can move on. But then, especially before the internet or before Google became what it is, and you can Google things and you can actually find information. But then I think that if you know that the end goal of that little experiment or that project could be to either identify a novel molecule, a novel drug target, I think it gives you purpose.
Sure.
It also allows you to look up and look at the broader picture and motivate you. And maybe you're lucky and you find something really interesting, but then you don't know what to do with it. And it's an opportunity missed for the person making the discovery to learn something and get out of their comfort zone and have an impact, but also from a patient's perspective. So I think it's a really, really nice, cool initiative.
I mean, you basically, you as a PI, I don't have the expertise to push this any further, so I'm just going to let it go and let somebody else do it.
Yeah.
This exists in order to catch those people and convert them to entrepreneurs in some way,
Yeah.
where they want to go that extra step.
Yeah.
Either to, either, they're not going to form their own drug company, okay, but they'll license it to somebody at a higher value because they've passed the bar for what pharma wants to see in target validation, rather than, you publish a paper and you do one little experiment and think you've validated your target, right? But up to the standards that a pharma company agree that, that would be a really cool target.
Absolutely. And I think I've always thought, well, actually always, I think about academia and industry as two sides of the same coin. Each side has its fortes. And I think both sides benefit from working together and collaborating. And that this is a great bridge or link between those two sides.
I think it's actually a little bit more nuanced than that. So having worked with basic science people and then running a basic science lab myself, but also working in a more medical environment, there are different styles of research. Even within basic research, there are different styles. So there are people, like me. I want to find a new molecule. I want to find a new enzyme. I want to find a new function. I want to find a new signaling pathway. I want, basic. And then there are people, particularly physicians, they're interested in the disease. They're interested in, I spent a lot of time doing liver fibrosis. Okay, so they're interested in liver fibrosis, and they're agnostic about the pathway, right? They'll do a Google search or a PubMed search. Oh, we should be thinking about, this molecule or this pathway this week. And then if that doesn't pan out, next week they'll have a different one. And that's a step toward what you do at a pharmaceutical company, but it's not really quite there yet. But that's a whole different style of research. Is, we're interested in an outcome, a process, a disease, and we're going to look wherever needs to be looked in order to find it, rather than someone who's like, I'm going to make my career by looking at this one GPCR and only the B subtype, not the A subtype, right? I'm going to look at this one, and just be hyper focused on everything to do with that. And they're very different, and they need each other.
They do. They absolutely do. I think that goes back, and the link between those two is the validated target.
Yes.
In a specific disease area. So if it's a GPCR, great. If it's not a GPCR, it doesn't matter because it's a validated target for that disease area, which I find really, really great. All right, so let's talk. We talked a little bit. Let's shift gears a little bit. We talked about you mentioned aha moments. And I have this question about the top 3 aha moments that you had as in your career that shaped your trajectory. Anything you'd like to add to the?
I mean, to me, it's not that I found a new thing necessarily, but that there's something like literally changed the direction of where I'm going. And I think, choosing what lab I worked for coming out of college made a world of difference, right? Because it got me involved in an environment where it was a very cutting edge of G protein research at the time, back before there were GPCRs, because we didn't, we kind of knew that there were lots of receptors, but there was only one G protein GS at the time, right? And so I got very lucky there, and that kind of set a trajectory that I've followed through my career. I'd mentioned cloning GRK5 accidentally, which was certainly it appeared at just the right time when the other project was fizzling to have it's like very clear. Okay, I know what to do with this. I'm gonna express it. We're gonna make antibodies. We're gonna characterize the enzymatic function. And then as people started making knockouts, in the Corona lab, said, we should make knockouts as well. And so that whole pathway led me in that direction, which was a lot of fun, but wasn't necessarily intended. Yeah. I think a third one would be just I was looking to see what things bind to GRK2, and I found this interesting ARF-gap protein that was affecting receptor endocytosis, but then it has a whole nother life. So we did a 2 hybrid screen with it and found that it interacted with another protein that kind of led into all the things that it does with at focal adhesions and in cell migration and all kinds of other physiological events that I would never thought that I would be interested in before I happened to do in that case it was a 2 hybrid screen, and that was the the hit I found, and that's what it turned out to be. Yeah. So
Wow.
that's like being open to what your results present you with.
Yeah, the next opportunities, which gets us into my next question very nicely, is what is the advice that you have for junior scientists who want to contribute to the field?
Yeah, there's this joke about the airplane with the holes in it, and and that basically here's a picture of an airplane that comes back to base, and it's got lots of holes in it, and those are the spots that are not important. So basically, someone who's old, giving you advice on how to start your career, I'm going to tell you what worked for me, whether it was accidental or not. And the world has changed. And to give you an idea of the way the world has changed, I think it's very important for people to think carefully and read broadly. And so one thing I learned very early, so the very first summer I was in research, the graduate students would take me down to the medical library at MD Anderson, and they had a table. And on the table, they put out the day's new journals. Whatever came in the mail that day was out. And I would just go and flip through the table of contents. And I was not that, I didn't knowledgeable then. I just had a high school education at the point. And I just started reading papers. And I would read all kind, everything, all kinds of papers. And one of the things I kind of, I regret about the way things are now is you get everything as a table of contents online, and journals I could beat them up. So they show you the title, and then they'll show you like 3 authors, and then it truncates. They won't even show you the whole author list. A lot of journals. So it's like they're people you'd read their paper if you knew they were on the paper, but you don't know. You just read the title. And there's a barrier now to clicking the link to I don't want to download a PDF just to glance and see if I'm interested. Right. And so that barrier didn't used to exist. You would have paper journals and you just kind of flip through them and see, right. And you see what's interesting out there. And now it's much harder for people to keep up. So I think, people, not in addition to there being a lot more to learn, because science is much more advanced than it was. It's harder in a lot of ways to keep up. So, I get emails of, the table of contents of various journals, but it's not the same as just, here's a random journal I wouldn't necessarily look at. I look at it, just kind of flip through and see what's interesting. And, people don't necessarily realize just how fast things have advanced. So I was very lucky when I was applying for postdoctoral positions. I applied and got a Helen Hay Whitney Foundation fellowship, which is based in New York City, and I was in New York City at the time. So I went down to Rockefeller, your stomping grounds there, to interview. And the guy who interviewed me was a guy named Macklin McCarty, who, if you remember your biology, in 1945 published a paper showing that tuberculosis bacteria were smooth or rough, blah, 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 blah. He's the guy who showed that DNA is the heritable principle. So
wow.
I mean, from my perspective, it's not that long ago.
Wow, that's impressive.
It was still active when,
yeah. Wow. Yeah, I think
it's got a lot more to learn. Sorry.
No, I think you make a great point about things being so fast, and the fact that we don't get the opportunity to actually appreciate all of these discoveries. I mean, every week, just to tell you, we pull down the latest publications for the previous week from PubMed, and we categorize them into different categories that we have determined. And these categories typically are the same, but if there's a collection of papers that doesn't fit anywhere, then we create a new category. And every week, just in the GPCR field, we have anywhere between 10 and 20 papers published every week.
Yep. But what you lack, and my most highly cited paper was a review that I wrote with Lefkowitz and another postdoc on 7 transmembrane receptors in Nature Cell Biology reviews. And this is from 2001, something like that. It is basically the last review that covers the whole field. So it still gets cited, a couple times a week. And if you want a review like that for G proteins, you got to go back to the mid 90s. There's a review by Gilman. There are no, there's like textbook material at this point, but it's not. If you want to dig in, you want a reference for your paper that really digs into the details. There are no reviews that, there'll be a review of, this receptor family or, but not the field as a whole.
You're right. You're right.
We kind of need, we need more updated versions of those, but wow, that'd be a chore to pull together.
We do. Not too long ago, I was putting together a very basic presentation on GPCR pharmacology, and I struggled to find reviews that were,
broad enough.
Exactly, complete enough, going, and there were, before the year 2000, after the year 2000, there aren't many, and it's very, very difficult. You could find videos that explain to you the basics. Or exactly, but still, it's not a publication, and it's very difficult. Very, very difficult. So I think I think it's a good way to spread the word out and tell the audience, if you're interested in writing something, that I think we need it. We need to sit down and write reviews and capture what has happened in the past 20, 30, 40 years, so that we can have it, and we can go back and appreciate all the advances that the field went through to get to where we are today. All right. So last thing, any other advice that you might have? I think the read broadly comes back to Paul Insel had the exact same advice. Read a lot, read broadly. I think what I typically have as well is, whenever you're looking into a lab, talk to people, talk to the PI, see what kind of projects they're available, or if you can bring your own ideas to the table, as you did with Bob saying, "Oh, I want to do this." Okay.
They might say no. They would, yeah.
Exactly.
I mean, the other thing you would want to, so I said think broadly, right? So you want to look at how the field is going, what looks like, where there are holes in the field. But just to reiterate, always have a backup plan. So if you're, have a main project that it really is pie in the sky, aiming for the fence, but always have something else going on, because things don't work. I mean, as I say, a lot of things fail. And sometimes it's individual experiments that fail, but sometimes it's the whole thing fails. And having something counts for a lot more than having tried and not made it. But also, being a bit unafraid to, look after yourself. So that can be everything from, cold calling, or, just cold emailing people, inquiring about jobs. Or looking around and seeing, if you're in a project and you're getting kind of stuck, and the assistance in your lab isn't quite helping, try and start your own collaboration. Talk to people outside your area that may be able to help you. people tend to get very passive and wait for their PI to do everything for them. And it's nice to loop them in, right? So I'm talking to somebody and then,
Yeah.
but you should have the initiative. It's your project. And I whenever even like a high school student, we get a new person in the lab, at their level, I tell them, okay, so you're an undergraduate, by the time you leave this lab, you're here for a couple of years, you'll read some papers, you'll get some experience. You'll go to a lecture in your med school class and you will know more than the professor on that little topic, but you'll know more, right? And your goal as a graduate student is to know more than your PI about the thing you're working on. You may not catch up to them on the breadth of what they know and the wealth of experience, but on the topic that you're working on, that you should be aiming for that.
Yeah.
Be
Absolutely. And this is exactly what I tell everyone, all the early career scientists that I invite to the podcast, who I have to beg to come and join on the podcast, because they come back and they say, well, I don't have 25 years of experience or 30 years of experience. And my answer to them is, no, you don't, but you are the expert at your own project.
That's right.
It is yours to become an expert at. You can speak about it. And to your point, it's your project. You should aim to be the expert, even have a better understanding and knowledge than your own PI.
Yes, but that's the immediate goal, right? Because in the end, right, all the skills I learned, as a graduate student and a postdoc, right, I did 100,000 at least Illumina and Dowex columns measuring cyclic AMP activity. Okay, no one does that anymore. I clone things. I have magic hands for cloning things. I can make huge constructs that people can't get. I can get them over. No one does this anymore. So the things that you learn and the things you're learning now, I don't know, Brett, everyone loves Brett, but who knows what the next thing is going to be? It may be obsolete. Whatever it may be that you become the expert at, you're becoming expert at that, but you're becoming an expert on how to become an expert. That's the really important thing, because the individual things that you learn, at some point they will be obsolete. And if you've learned how to learn and you're adaptable, you can keep up and you just go, as new techniques become available, you learn them too.
Exactly. I think that's also a great point, is that a lot of times people tell me, oh, you mean I don't want to apply for this job because I don't have this and this and this skill, but I do have this and this and that. And I say, well, look, you've learned skill A. What are the things in common with skill B that you can highlight and show that you learned it quickly or efficiently, or you published? And it's all about how adaptable you are and how you can use your learning ability to learn new things and to adapt. The other thing that you mentioned about collaborations, I want to say I was a naive graduate student who thought that I had to do everything on the project so that I can get the paper published. And later on, towards the end of my PhD, I realized, well, this is never gonna work. And that's when I realized that actually you don't need to know how to do everything. You need to figure out what is it that you need to do, figure out how long it will take you, and figure out who are the best people who know some of these things, and go and collaborate with them, and then direct it. Because the goal is, obviously you have to develop your skills and know how to set up experiments and run experiments. But at the end of the day, the goal is to help answer scientific questions.
Right.
And by the time you're done, you've managed a project, you created a project, you strategically build out an entire process. You wrote, you read. Exactly. And all of these skills are very important. And yes, as you mentioned, running, purifying radioactive cyclic AMP to then measure cyclic AMP production, or inhibition of cyclic AMP production, becomes obsolete. But the process around building something will always be a great skill to have.
Yep.
And with this, Richard, thank you so much for your time.
Well, thank you.
And I hope, we'll talk to you soon, and we'll bring the audience a really neat series of podcast episodes about the history of our field.
A lot of cool stuff.
I think so too. I think so too. Don't go anywhere. We're going to let people wonder what we talk about. And with this, our episode has ended. Thank you. Can I ask you for a favor? Please subscribe to our Dr. GPCR YouTube channel. Many of you come back regularly to watch our videos, like the monthly video edition of the newsletter, but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank you for listening, thank our guest for joining us, and also would like to take a moment to thank our team members, Attila, Ines, Montserrat, Ivana, Andreina, and Balint. A huge thank you to our Dr. GPCR Ecosystem partners, Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. If you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. You can also subscribe to our monthly newsletter delivered directly to your inbox by visiting ecosystem.drgpcr.com/newsletter-sign-up. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.